Hello everyone, welcome back. So let us continue looking at our series of lectures on carbohydrate metabolism. And today we're going to look at the effects of pyruvate. The last time we saw glucose was broken down in glycolysis to pyruvate. So in this particular class, we want to answer the question, what happens to this pyruvate? Because we do know carbohydrates are completely broken down to carbon dioxide and water. We know the electrons are transferred to oxygen in the electron transport chain to make water, and in the process we generate ATP. But what happens to pyruvate? Although we have broken down the glucose molecule into 2,3 carbon molecules, we have not yet seen carbon dioxide. So the three effects of pyruvate we're going to look at are lactate, ethanol, and acetyl-CoA. So the effects of pyruvate depend on whether the cell is metabolizing glucose in the presence or in the absence of oxygen. So oxygen is very fundamental in determining the effects of pyruvate. So in the absence of oxygen, we call this anaerobic conditions. And so under anaerobic conditions, two things can happen to pyruvate. One, it can be converted to lactate, or it can be converted to ethanol. So in lactate, here is catalyzed by an enzyme called lactate dehydrogenase, and we're going to see a bit more detail in lactate formation. In yeast and some bacteria, pyruvate is converted to ethanol, okay? And this reaction is called a fermentation, and it is what is commonly used to make alcohol, and we're going to look this in a bit more details in the following session. In the presence of oxygen, which we refer to as aerobic, pyruvate is converted to acetyl-CoA. So we know glycolysis occurred in the cytosol. And so in the presence of oxygen, pyruvate is transported in the mitochondria, where we have the pyruvate dehydrogenase complex, which is involved in converting our pyruvate into acetyl-CoA. We are also going to look at this in more detail. So we say the first phase of pyruvate is conversion to lactate. And we say this occurs in the anaerobic environment. So in glycolysis, if we look at the substrates that are required, one of them was the oxidized form of NAD, NAD, and glucose. And the end products, besides ATP, we also required, we had the reduced form of NAD and pyruvate. So we do know in glycolysis, if pyruvate and NAD accumulate, it will prevent the reaction moving forward. So in the absence of oxygen, how do we ensure? Because normally the NADH is taken to the electron transport chain, which only occurs in the presence of oxygen. So in the absence of oxygen, how can the cells make sure glycolysis will continue? So what happens is that pyruvate is converted to lactate. Now, in this reaction, we can see the pyruvate that is accumulating in the cytoplasm will now be converted to lactate, and we're going to have reduction of pyruvate. So that makes sure pyruvate does not accumulate, making sure there's a low concentration of pyruvate so that glycolysis can continue forward towards its end product. The other problem we had was NAD, okay, the reduced form of NAD. So what happens is that these electrons that are carried by the reduced form of NAD are added to pyruvate at this point, we can see on this second carbon, and the NAD is oxidized back to its NAD plus, the oxidized form, while pyruvate is reduced to lactate. So lactate is carrying the electrons, and it serves both purposes of reducing the cytoplasmic concentration of pyruvate and reduced NAD or NADH. Okay, so this is the first phase of pyruvate, and this occurs in an anaerobic environment. And where we can find an anaerobic environment is when we have vigorously contracting muscle. So this muscle is strongly contracted, and it closes the blood vessels that bring in oxygen. So at that moment in time, it's operating in an anaerobic environment. We also mentioned in cancer cells, if you look at the previous lecture on glycolysis. There are some tissues also that can convert pyruvate to lactate even in the presence of oxygen. For example, the red blood cells or erythrocytes and retina. In the erythrocytes, we know we don't have the mitochondria. The cells are lacking organelles. So the only way they can catabolize glucose is through pyruvate, and then pyruvate is converted to lactate. So what happens to this lactate? Like, for example, in the erythrocytes. So in skeletal muscle and red blood cells, we can see our glucose is broken down to glycolysis to pyruvate, and the pyruvate is converted to lactate. We have discussed that previously. So what happens to this lactate? For example, in muscle or red blood cells, the lactate is carried in blood back to the liver. And in the liver, the lactate can be converted back to glucose through the process of gluconeogenesis. We're going to look at this process, the synthesis of glucose from non-carbohydrate sources like lactate. So this cycle where the lactate is carried from the muscle to the liver, converted to lactate, then lactate is used to synthesize glucose, and glucose is carried back to muscle or the red blood cells to continue with the production of ATP, is what we refer to as the Cori cycle. And the Cori cycle predominantly operates between the skeletal muscle and the red blood cells and the liver. So in summary, glucose is broken down to pyruvate in these tissues, muscle and red blood cells. Pyruvate is converted to lactate by the lactate dehydrogenase enzyme. And then in the liver, lactate is converted back to pyruvate. You're going to see this by another isoenzyme of lactate dehydrogenase. And pyruvate is used in the process of gluconeogenesis. And the glucose formed can be carried back into the bloodstream and carried back to the red blood cells on the skeletal muscle. The next phase of pyruvate is formation of ethanol. So this, as we've mentioned, it occurs in some plant tissues, in invertebrates, and in the yeast. In the yeast that is used to form alcohol, is commercially used to form alcohol. So this is a two-step reaction where first pyruvate is decarboxylated by the pyruvate decarboxylase enzyme. So in decarboxylation, we lose one carbon, carbon dioxide is lost, and we end up with a two-carbon compound of acetaldehyde, as you can see here. The next reaction is the reduction of the aldehyde group in acetaldehyde, and this reduction means addition of hydrogens or electrons, and these electrons are added to the aldehyde group to give us our alcohol group, and we form the compound of ethanol. So this particular second reaction is carried out by an enzyme called alcohol dehydrogenase. And we can see it uses NAD. The reduced form of NAD is the donor of these electrons to acetaldehyde to form ethanol. So if we think about it the way we're talking about lactate dehydrogenase enzyme, similarly, the purpose of this reaction is to allow glycolysis to occur in these cells, because we'll see pyruvate is reduced, and also the reduced concentration of NAD, reduced form of NAD will be reduced. So the end product in this particular reaction will be ethanol. This reaction is of particular interest in us because we have alcohol dehydrogenase enzyme in humans. However, in humans, the alcohol dehydrogenase enzyme does not make ethanol. However, it does the reverse reaction, where it breaks down ethanol to acetaldehyde. Okay, so as we said, alcohol dehydrogenase is predominantly found in the liver, and it converts ethanol back to acetaldehyde. Now, acetaldehyde is toxic, is a free radical, and it can cause damage to DNA and also the cells, cause damage to the cells. And in this case, it will be causing damage to the hepatocytes or the liver cells, to the liver cells. So if we have high amounts of ethanol in the body, normally what happens is this acetaldehyde is converted by acetaldehyde dehydrogenase, this enzyme, to acetate, and acetate is converted to acetyl-CoA, which can be used to produce energy in the TCA cycle. Okay. However, if there's high amounts of alcohol consumed, we have the formation of acetaldehyde can be much faster than the function of aldehyde dehydrogenase enzyme, and this high concentration of acetaldehyde is the ones that damages the liver and increases the risk of cancer because of increased damage to DNA. So this acetaldehyde is the one that causes liver cirrhosis and increases the risk of liver cancer. We do know there are people who have different tolerance to alcohol. Some people need to drink a lot and what, and all these are come about from genetic variations in alcohol dehydrogenase enzyme and aldehyde dehydrogenase enzyme. So we can see we have seen alcohol dehydrogenase are being used in yeast tissues to form ethanol, but it's also present in human liver where it is used to convert ethanol to acetaldehyde. Okay, so this is an important clinical correlation of alcohol dehydrogenase. So in presence of oxygen, we saw pyruvate will be converted to acetyl-CoA. So acetyl-CoA is formed in aerobic organisms and it's catalyzed by an enzyme called pyruvate dehydrogenase. We're going to look at this enzyme. And this is not just an enzyme, it's called pyruvate dehydrogenase complex because it is made up of 3 enzymes. We're going to look at them and 5 coenzymes. So in this particular reaction, acetyl-CoA will be used in the TCA cycle. But let's look at what happens to pyruvate. In this reaction, pyruvate is decarboxylated, and we can see we lose carbon dioxide. So it was a 3-carbon compound, and we can see we are losing 1 carbon as carbon dioxide. And our pyruvate, the 2-carbon compound, is now attached to coenzyme A to form our acetyl-CoA. Okay, so remember we had said at the beginning, carbohydrates are broken down to carbon dioxide, and now we can see here our first carbon dioxide coming from pyruvate. So this particular enzyme allows efficient direct transfer of the product from one enzyme to another. Let's look at pyruvate dehydrogenase in a bit more detail. So the pyruvate dehydrogenase reaction will occur in the mitochondrial matrix, which is the location of the pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex, as we had said, is a complex because it's made up of 3 enzymes. The first enzyme is pyruvate dehydrogenase, or E1, and it is bound with the cofactor coenzyme thiamine pyrophosphate, or TPP. So E1 catalyzes the decarboxylation of pyruvate to acetyl. So it removes the carboxyl group from pyruvate to give us our acetyl group. E2, also called the hydrolipol transacetylase, takes the acetyl group from E1 and adds coenzyme A to it to give us our acetyl-CoA. Okay, so this is the function of the E2 enzyme, and it has the 2 coenzymes, lipoic acid and coenzyme A as their coenzyme. So remember lipoic acid, we saw it has a sulfur group, sulfhydrid group, and this sulfhydrid group, sulfur groups are usually reduced to form the disulfide bond, which will now be reduced by the 3rd enzyme E3, which is called the hydrolipoyl dehydrogenase. So this 3rd enzyme, the hydrolipoyl dehydrogenase, regenerates the lipoic acid, which had been reduced by the activity of the hydrolipoyl transacetylase. And these electrons that come from the reduced lipoic acid are carried through FAD and eventually end up at NAD, so that we have one of the end products of the pyruvate dehydrogenase reaction as the reduced form of NADH plus H. So these are the 3 enzyme entities of pyruvate dehydrogenase complex, E1, E2, and E3. In addition to the 3 enzymes, we know pyruvate dehydrogenase requires 5 coenzymes. The first one we saw, thiamine pyrophosphate, which is derived from B1 or thiamine enzyme, is very important for the decarboxylation of pyruvate, and we see it's a coenzyme and sits at the active site and actively participates in the decarboxylation of pyruvate. The other coenzymes we have seen is FAD, which is derived from riboflavin and serves as an electron carrier in the pyruvate dehydrogenase reaction. We have also seen NAD, which also serves as an electron acceptor in the pyruvate dehydrogenase reaction, and it accepts the electrons from FAD, which are coming from lipoic acid. We have also seen coenzyme A, which is a vitamin derived, which is derived from pantothenic acid, and it forms acetyl-CoA, which we will see is a very key molecule for the TCA cycle. Then we have lipoic acid, which is not really a vitamin. It's not directly a vitamin, but we have seen it acting as an electron carrier in E2, and also an acyl transporter within E2 complex, E2 enzymes of pyruvate dehydrogenase. So this vital role of the vitamins in the pyruvate dehydrogenase enzyme are very important in energy production, especially in the central nervous system, where we see when you have deficiency of B vitamins, we have some neurological symptoms associated with some of these deficiencies. And we can see because B1, B2, and B3 play very important in functions in energy production. And we know the brain or the central nervous system predominantly utilizes glucose for its energy production. So this deficiency of these vitamins will impact the function of pyruvate dehydrogenase, and in turn will impact brain function, brain and nerve function. Also, insufficient coenzymes in the pyruvate dehydrogenase complex will lead to reduced aerobic ATP, and this will present with symptoms such as fatigue and reduced exercise tolerance. So we can see vitamins are very important for the functions. These vitamins are very important for the functions of pyruvate dehydrogenase and in turn energy production. So how is the pyruvate dehydrogenase complex regulated? We can think about it in 4 ways. So first we have the allosteric regulation, and we mentioned yesterday an allosteric enzyme has multiple subunits and has several binding sites in addition to the active site. So we have inhibitors of the pyruvate dehydrogenase complex. For example, because pyruvate dehydrogenase serves to produce energy, so high energy levels indicated by ATP, NADH, and acetyl-CoA will inhibit the pyruvate dehydrogenase activity. So these are negative modulators of allosteric of pyruvate dehydrogenase. Then we have activators, which are low energy level indicators such as ADP, the oxidized NAD, and pyruvate. All indicate high energy demand and promote the pyruvate dehydrogenase activity. So this is moment to moment, depending on what is present in the cell. Then we have covalent modification. So covalent modification is a form of enzyme regulation where a group is added covalently to the enzyme, and this either activates or deactivates the enzyme. For example, you have an enzyme called pyruvate dehydrogenase kinase, which is able to phosphorylate the pyruvate dehydrogenase complex, and this activates the pyruvate dehydrogenase, inactivates it when the ATP levels are high. So if we phosphorylate pyruvate dehydrogenase, we inactivate it. When the ATP levels are low, we have the enzyme pyruvate dehydrogenase phosphatase, which dephosphorylates pyruvate dehydrogenase complex and in turn activates it. So this dephosphorylation will happen maybe in presence of signals like insulin, which stimulate the breakdown of glucose. So we have hormonal regulation, mainly by insulin, which stimulates pyruvate dehydrogenase activity in tissues like muscle and adipose tissue. And this promotes glucose utilization and fat storage. And you can see the way it does this is by promoting dephosphorylation of pyruvate dehydrogenase enzyme, which will activate it. Then we have, interestingly, calcium. So calcium plays a very big role in muscle contractility, especially skeletal muscle contractility. And we see calcium activates pyruvate dehydrogenase complex in muscle cells during contraction. And during contraction, we know that we have a high demand for ATP for contraction. And so it's only fits the story when calcium activates pyruvate dehydrogenase, which is involved in formation of ATP. And so in this particular lecture, we have looked at the effects of pyruvate, what happens in aerobic condition and anaerobic condition. Thank you so much for your time.